Well, I'd like to welcome members of the NJMA and the Boston Mycological Club and the Eastern Pennsylvania Mushroomers. It's great being able to share speakers with such a big, diverse, terrific group. Um, I'm really excited to have James Cameron Mitchell with us tonight to talk about these Resinicolous fungi. James is a physics graduate student at Harvard. He started his graduate research focusing on the nervous system development of roundworms. And at Harvard, James became involved with the BMC, and it redirected the focus of his interest. And now his research is concentrated on these fungi that live on this inhospitable environment of conifer resin. I'm sure that many of you are familiar with James from using iNaturalist. He's probably the most active curator of fungi on iNaturalist. So if his name isn't familiar to you, it will be now. And I'm really excited to have James talking about this really fascinating group of fungi that are really, a lot of them are really, really common since they came onto my radar thanks to James about a year ago. We found a number of them in New York City, and it's exciting to the prospect of different mushroom clubs having an eye out for these really incredible organisms. So with that, I hand it over to you, James. Welcome.
Thank you. Thank you. Yeah, my name is James Mitchell. I'm here to talk to you about really one of my passions, Resinicolous fungi. So what I mean when I talk about resinicolous fungi is these fungi that grow on resin. And you might ask what resin is. You probably all have some sort of understanding of what it is. Tom already mentioned, really, I'm going to be talking about probably this gymnosperm resin, this stuff on pines that you see. But how resin is defined technically is that it's a lipid soluble compound or mix that is produced by plants. It's usually of terpenoid and phenolic compounds. Generally, it's secreted in specialized structures, and it might have some ecological function, either to protect trees or just even cover wounds. So it's not, this class of substances are not just produced by things like pine trees, and quite a number of angiosperms do produce resin. And in fact, there are a handful of fungi, mostly kind of tropical, that live on angiosperm resin. But ultimately, we're not really going to talk about those. We're really just going to talk about the fungi that grow on gymnosperm resin, because there are more of them. They're somewhat better characterized, but I think probably by the end of the talk, you'll be able to take away the fact that they're really not that well characterized either. So mostly where you're going to find resin in terms of conifers are things in Pinaceae. So larches, spruce, firs, pines, hemlock sometimes. Another big family for keeping an eye out on if you have them in your area, which obviously they're not native to here, is Oricariaceae. So these southern hemisphere sort of tropical conifers generally tend to produce a lot of resin, and really these haven't been worked over a lot, but like I said, they don't live here anyway. But then also the Cupressaceae. So the family that contains things like junipers and Atlantic white cedar and northern white cedar. They also produce resin, and there are some resinicolous fungi on them too, though generally fewer, because they don't tend to produce a lot of resin, but they're worth a look. So if we look at the study of these fungi that live on this substrate through history, really this started actually almost at the birth of mycology itself with one of the fathers of mycology, Christian Persoon. And the fungus that he described in 1801 is actually one I'm going to talk about later in this presentation. But basically since then, this has occurred in drips and drabs. People haven't really worked over this ecological niche in great detail. It's just been done in bits and pieces. One exception to this was an Italian in the 1920s who published a few small papers about basically all the fungi he could find that grew on resin in his area. But he's really an exception, and the papers are pretty obscure. So in the modern era, there's a group of fungi really that's been worked on a lot, and the person describing most of them on resin has been this man, Juuko Rikkonen, and also his collaborators. And we'll talk about that group a little bit later too. There should be some species in the Northeast, generally a little further up north. But this is where most of the work has been recently. And at this point, there are about 49 published species of fungi that grow on resin. So if we look also at all of these resinicolous fungi, we might also ask, like, where do these fall in the tree of life? Because the fungal tree of life at this point is, pretty broad. There are a lot of different groups, but it turns out, with few exceptions, these all fall into the group traditionally thought of as sort of the higher ascomycetes, Pizizomycotina. So these sort of filamentous ascomycetes. And if we look even further in there, we see really actually there's a fairly wide occurrence of these fungi. We've got a big concentration in the class Uroshiomycetes, which tend to be kind of oddball fungi. You have things like Aspergillus and what else? Aspergillus and similar fungi. Onagina is also in Uroshiomycetes. We also have a few members in Leoshiomycetes, so the inoperculate cup fungi. A couple things putatively in Sordarioomycetes, one of the groups of Pyrenomycetes. Dithydiomycetes as well, so another group of Pyrenomycetes, the bitunicate ascomycetes. And also published very recently, really just last month, about 7 species in Orbiliomycetes. And additionally, a fair number, again, this is something I'll talk about kind of at length later, in a relatively new class called Sariomycetes. There might also be some species in Lecanoromycetes, which are where most lichens are found, but there aren't really any published species confirmed in that class at this point. So if any of you are curious to find a list of these things, I keep a relatively comprehensive list of published species on iNaturalist in my project Resinicolous Fungi. So the list of taxa eligible for inclusion in the project is a fairly good list of the Resinicolous fungi currently described. So now I'm going to try to focus generally through this talk about or on species that you might actually find in the northeastern United States or northeastern North America. And at 2 points with 2 species or 2 groups of species, I'm going to dip into work that I've done a little more in depth. And then for what time we have left, we can go over some interesting exotic species that haven't been reported either in this part of North America or in North America at all that are worth looking out for, because really this isn't a, this just isn't a substrate that's been looked at a lot. So I'm going to start out in Uroshiomycetes, because this has the biggest concentration of species. And one species that you might find in this area is a pretty recently described species, described by Keith Seifert and Joey Tanney up in Canada and from New Brunswick, but which is probably found on spruces, not only in the Acadian forest, but possibly also in Maine, probably further down. Which is a hyphomycete called Cerosabe oblongospora. So it forms these cinemata, these little sticks, and at the tip it produces these chains of dark hyphae. And like I said, this is pretty recently described, just last year, so far only known from New Brunswick, but it's something to keep an eye out for. And the other group in Uroshiomycetes, which is the most diverse group, is Mycochlyciales. So this group is globally distributed. So there have been species described from the Northern Hemisphere, the Southern Hemisphere, from North America, from Asia, from Europe. And generally these tend to grow on pretty old resin. But so far there are 19 species, of which at least 7 species should occur in northeastern North America. So generally these will all look fairly similar. Again, these look like sort of these pins with black or whitish pinheads, and they might have colors. So this one in the slide at the moment has green coloration on the stalk. But instead of being a hyphomycete, this time these are actually apothecia. So they're just apothecia with really long stalks. And this is a pretty interesting group because it's generally been, well, historically overlooked, but they're also really big in use as indicator species for old growth forest status. So in part, that might be why so much focus has been put on these in recent years. So this is one of the species that we could find in at least Acadian forests. I think this is Chenothecopsis cladinii, which I believe occurs on spruces. Here's another species, Chenothecopsis marcinii, which occurs on, I believe, hemlocks. So these are, as the scale bar suggests, really small. So this is maybe a 1/4 of a millimeter tall. And another complication with finding these, so says one of the people who looks for them, is that instead of really occurring on the outside of the wood, they tend to occur in sort of hidden resin pockets inside. So they're difficult to find. So they're really underreported because you really have to dig through the wood to find the pockets where these things are even growing. But it would be exciting to see some more reports of these, especially from northeastern North America. Another interesting fact, part about these species is that these are one of the few fungi that are actually conclusively present in the fossil record. And you might guess where or what type of fossils you'd find. something that grows on resin. And if you guessed amber, you'd be right. So there are several examples of these caught in amber, and really these are pretty amazing fossils because the preservation is amazing to the point that you can even see septate spores drifting off in the resin. So several of these have been described, mostly from amber deposits, I think in Europe. And these, as in most of the other groups we're going to talk about, really, as a group, are not solely resin dwellers. There are many members, 19, that grow on resin, but there are also members of this group, this genus, Chenophycopsis, that grow on lichens as parasites. There are ones that just grow on dead wood, and there are ones that are lichens themselves. So this is another sort of interesting fact here, is that a lot of these Resinicolous species don't seem to come from solely Resinicolous lineages. These seem to have arisen among other lifestyles, just as another lifestyle that one can have. So another species that I think is actually pretty common in the Northeast, especially I think in high altitude areas, but is overlooked, is in Dithydiomycetes, a fungus in the genus Strigopodia called Strigopodia resini. So if any of you have been, say, high up in the mountains where there are firs and spruce, sometimes if you look on resin, you'll find very dense velvety mats of hyphae, just like really dense black velvety mats. And sometimes if you poke around under the surface of the mats, you can find little parathecia. And these are the parathecia of Strigopodia resini. You can also find in among the hyphae, you can find conidia, which are these long sort of helminthosporium type conidia. So brown, septate, very long, slightly falcate. But this is a pretty fun species. I've seen it a couple times, mostly from older barium specimens. But you can often find the conidia being produced directly off of ascospores. So it'll produce these little phyllids and start shooting off conidia. And it's really a fun find, but again, generally underrepresented, I think. So at this point, I'll also say that for many of these species that I'm presenting, there are other relatives in other parts of the world. So for this one, there's another species, Strigopodia batiste, that is found in the Pacific Northwest that looks almost exactly the same, but grows on Douglas fir. But yeah, there really hasn't been much work done on these at all since they were described in the 50s and 60s. So it would be interesting to have more observations of these. The next group, and a pretty common one actually in Dithydiomycetes, is these Resinicolous species in the genus Mytilenidian. So some of you might have seen Mytilenidians before. They have brown septate spores. They generally look like little mussels, little black shells that have been stuck to the substrate that they're growing on. This is a pretty good illustration of Mytilenidian resinicola from the original publication in 1933. And this is one of these species that should be present around here. Again, not very frequently reported, but I've definitely seen specimens from at least Maine. There's specimens from Michigan. So this should be around. And while there's one published species, Mytilenidian resinicola, there also seems to be an unpublished species with less constricted septa in the spores and with slightly shorter spores that's also present in this area. But doesn't seem to have ever been described as far as I can tell. For a better picture of it, you can see them here. Obviously in the field, this is going to be pretty hard to point or to see. So in this photograph, I think there are 2 ascomata, one here, one here. And in this specimen, you have one growing here and one growing here. So not the easiest things to pick out while you're out in the field, which is generally true of these, especially the little dark things. They're just not easy to see. So for those of you with keen eyes, in this second photograph, you might have noticed that in the center, there seemed to be maybe some apothecia as well. And if you guess, you'd be right. So this is another one of the Resinicolous species that's found in our area. And this one I found a little further south, so I've definitely found this in the Boston area. And if I found it here on relatively young pines, I don't really see why it shouldn't be further south, which is moving to Leoshiomyces, cup fungi, the species Clausinomyces olivaceus. So these apothecia tend to be green. Specimens from North America, the hymenium seems to be pretty shiny usually, whereas other things have more granular dull hymenium. These have a pretty shiny hymenium usually. And these have a really interesting characteristic that not many fungi have, that they produce 8 ascospores, which then inside the ascus, before it shoots spores, each produce a ball of conidia. And really the conidia are what gets shot out. And there's another graduate student, or sorry, there's a postdoc here at the Farlow who studies these specifically, Luis Quijada. So this is one of the groups that he really works on. But yeah, this is another species that really you should be able to find in our area. And I've found it a few times in Massachusetts. Another one that should be really pretty commonly findable, and I've heard Tom at least say that he's definitely found it in New York, is Lachnellula resinaria. So this is, I mean, compared to most of the ones I've showed you so far, this looks a lot more charismatic, right? You've got bright colors, you've got this like white furry and this bright yellow hymenium. And this you'll find generally on a bunch of different conifers growing on the resinous exudates. And it's characterized well by having really small spores. So in the range of, I think, about 4 microns long, but still elliptical, not really globose spores. But yeah, this is one of the things that should be relatively common. Often when they're dry, they're really curled up and you can only see the white outside, not really the yellow hymenium. But if you give them a good spritz of water, they'll revive up and you can see them. And it's a really pretty, really standout species. Now another species, and one that I've actually worked on a little bit, and I'll talk about that a little more in depth, is a species that is commonly called Bisparella resinicola. So this, for most of you thinking about Bisparella, this looks about right. It's a bright yellow orange thing. Resinicola, it grows on resin. Sounds like a great name, right? So this species is found in North America and also in Europe. In Europe, it's really only found in the Alps. In North America, it's found in the Pacific Northwest up to Alaska. It's found in down in like the Smoky Mountains National Park. It's found in New York. It's found in Maine. It probably just is wherever there are spruce trees and fir trees, really. So this kind of got me interested because this is a really broad distribution in North America, whereas in Europe it's really just stuck in the Alps. And another interesting thing is that this sexual stage with the apothecia is pretty rare. Really, there are only a few known samples from this area, Maine mostly, also the Pacific Northwest, really rare in Europe. And really what you're more likely to see rather than these little orange-yellow apothecia is you're more likely to see the anamorphic stage, which looks like little yellow matchsticks growing on resin. Like I said, definitely in Maine where there are spruces. Peck described it from the Adirondacks in New York. I'm like 150 years ago, so don't know if it's still there, but it would be worth looking. So yeah, just not a lot of reports for these fungi. And one thing of note that no one yet has explained is that you will never find it growing on pine. It has never been recorded growing on pine. It's been recorded on pretty much every other conifer in Pinaceae. So I think it's growing on hemlock, it grows on Douglas fir, it grows on larch, it grows on spruce, it grows on fir, but just not pine. So I actually, through iNaturalist, found somebody in Alaska who had collected this fungus. And he sent me a sample. And I already had some samples that that I had collected, or that some people I knew had collected up in Maine. And I managed to get some samples from Switzerland and sequenced them to try to find out like what's really going on here. And one of the questions I really wanted to answer was, is this really a Bisparella? Because really, when people talk about Bisparellas, it's a group of fungi that generally tend to be bright, kind of yellow, cup fungi, but otherwise not really pretty well defined as a genus. Really, a lot of things have been thrown together that maybe don't really seem too similar sometimes. So we sequenced these, and working with Peter Johnston in New Zealand, who has been doing a lot of these big phylogenies of Leoshiomycetes, we were able to get pretty good phylogeny. So I know this is really small, so I'm going to point out some salient features of this tree, which are that the type species of Bisparella, the real Bisparella, is in Holotiaceae. It's really closely related to Hymenocyphus, actually. But once you start to look at the other things that people usually think of as typical Bisparellas, so Bisparella citrina, for instance, or Bisparella sulphurina, mostly southern hemisphere, but also northern hemisphere as well. These species aren't even in the same family. These are often a different family, Pezizelaceae, and these are better thought of as members of the genus Calycina, which was actually an argument that was made 7 years ago by a German mycologist, Hans-Otto Borel, just based on morphological characteristics. He said that really these don't really look like they're actually related to real Bisparellas, but they are actually related to this genus Calycina. So the other result from this was that our fungi, this Bisparella resinicola, isn't actually related to either of these groups. So it's off on its own in Hyalocyphaceae, actually. And so the name really for this fungus is one of the oldest names. The epithet comes from the name that Pursoon gave it in 1801. And the genus comes from another famous European mycologist of the 1800s, Robinhurst. So the name for this fungus is Eustilbum aureum now, which is already published, already exists. And another takeaway from it is that even though we have these very strange geographic ranges ranges and these really big differences. Like it's really restricted in Europe, it's really broad in North America. As far as I can tell so far, it really looks like it's all the same species. It just seems to be one species everywhere. So there might be more work that needs to be done for this. The sampling is a little sparse, partly due to the fact that people hardly ever collect this fungus, but it would be, I think, really good for people to keep an eye out on it. So one of the interesting things in the course of this project is that I think I'll be the first person to realize that this exists really far south in the Appalachians, like in the Smoky Mountains National Park. And it's not because I've collected for it there, it's because I've seen it on other people's collections where it seems to have gone unnoticed. So this is probably out there, but people either don't know what it is, these yellow matchsticks, and think that it's another fungus, or think that it maybe is a lichen. So it would really be good to have people out there looking for these things. So the next group that I can pretty much definitely say that you can find almost anywhere that there are conifers, wherever you are in the Northeast, these are probably not more than a couple miles from you, are fungi in this group, Sareomycetes. So I mentioned this is a relatively new class. So it was erected last, I think, February by some collaborators and I to accommodate this little group of fungi. In our analyses, really, it just doesn't fit with any other group of fungi. Either just, it's traditionally been placed in like Anoromycetes with the lichens. It looks a lot like a lichen, but there's just no relationship there. It just doesn't come out in the molecular data. But similarly, there just doesn't seem to be a close relationship to any other known group of fungi. It's definitely a Hyraskomycete. It just doesn't have a close relationship to anything so far. So traditionally, this group, Sareomycetes, would be represented by one genus, Saria, and two species, both of which are illustrated here. So you have the black species here, nice little black apothecia, kind of granular looking on the surface, which is Saria deformis. And then you have this orange species, which is a lot more noticeable, frankly. Orange is just a lot easier to see on dark resin than black. And this species is Saria resini. And these species also have anamorphic stages. So Saria deformis has little black pycnidia. You might be able to see it in this picture. It's a little hard to see. It's not this one. This is just an apothecium. It's like this dot over here. So just really small black pycnidia, which are just these almost like parathecia, but like an anamorphic parathecium. So conidia inside, not asci. And Saria resini produces similar structures, orange, usually with little beak on them, and often you can see just masses of conidia, these white masses of conidia pouring out the top. And one interesting thing that'll come back a little later is that historically, these two groups, the anamorphs of these two fungi, were placed actually in different genera. So not even in the same genus, even though the teleomorphs were considered to be in the same genus. So this group is, like I mentioned, super common. You can find them pretty much anywhere there are conifers. They've been reported from North America, they've been reported from Europe, they've been reported from Africa even. They've been reported from Asia. They've been apparently introduced to New Zealand on the imported conifers that they grow down there. One strain of what would typically be called Saria deformis was even extracted from wood from one of the original huts built in Antarctica. So it was brought over with the wood to build the hut, and it apparently has just stayed alive in the wood there. So really common fungi. If you're anywhere with something in the pine family, it's probably there somewhere. But it's not just on the pine family. You can also find these on, especially in the Northeast, in Atlantic white cedar swamps. They grow really well on the resin of Atlantic white cedars. I've found them on junipers around here. I've seen at least one sample from Minnesota on northern white cedar, not a very common tree wild here, but yeah. And they've also been reported from like Japanese cypress, the Hinoki cypress, and also I think cryptomerias as well, which would again be an introduced tree here. So just a really common group of fungi, found on all sorts of hosts, found all over the place. So this made me very curious because this is sort of unusual. So when you look at things, usually they don't have so broad a host range, and usually they don't have so broad a geography without, in the modern day at least, people starting to find that really these aren't actually just single species, these are multiple species. So a few years ago, after I think Larry Millman first introduced me to these fungi, um, I really started looking for these, and I collected a bunch in this area. I got herbarium samples. I went on a collecting trip to California, all over the state to collect them. I had collaborators in Europe start sending me specimens that I sequenced. I started getting things from Asia, not too many, but some. And I sequenced these all and I started looking at them with my collaborators, one of whom was Luis Quijada, again, the postdoc here at the Farlow, who's been an important part of this project, and Don. And this group is, again, really diverse, actually. What looks at first glance like two species is, in the case of the orange one, maybe a dozen species or more that are really hard to separate out based on morphology. In the case of the black one, probably at least 3 species. And at this point, I've also come to the conclusion that really, instead of thinking of these as single species, probably this group is better seen as 3 different genera. So there's one genus around Sarhidiformis, the black species, and all those look pretty similar. There's one genus around Saria resini, the orange species, and then there's one completely novel genus that's actually not in northeastern North America. So far, it's only known from the Pacific Northwest and also one sequence from Northern Europe. So again, I know this tree is pretty hard to see. So let's zoom in on the 3 different chunks that I'm talking about here. So if we look at just the group around Saria deformis, which will be the real Sarias, we can see that even here, so I've been able to split this sort of morphologically into 3 groups. Even here, if you look at this tree, you can see that there's a lot of structure here. And if you were more inclined to splitting than I am, you might even say, oh, well, this looks actually like maybe it's even 5 or 6 species. But one of the problems that we've had here is that these are so common and they don't really seem to mind growing next to each other. So you might find some nice little black apothecia of Saria deformis and think that you have just one species. But then if you poke into it more, you might find that actually on this same specimen, you have multiple of these species growing right next to each other. So this has made sort of interpreting these sequencing results and lining them up with specimens pretty difficult, because most of the sequencing was done back at a point where I didn't realize that this would be the case, essentially. So I took an apothecium, sequenced it, and then later came back and realized that there are possibly 2 species here, and which is the one that I sequenced. So it turns out, though, that there is at least one group out of these 3 that is really easy to identify. And it's easy to identify because once you look at it under a microscope, the hymenium actually has this purple pigment. It's this beautiful purple color, pretty dark purple. And when you add KOH, it just goes blue. So it seems to be an indicator pigment that in a neutral or a basic environment turns blue. So this is a great feature to identify, right? Because this is just really easy to do. You look at it, it's purpley, maybe it's really faint, you can't really tell, but you add KOH and it goes this brilliant blue color. So that's great. And it's also great because this seems to be the real Saria deformis, because the type specimens that I've examined have this feature. What's not so great is that all the other ones just don't. And microscopically, the features that these have, the way the assayi look, the way they stain, are almost identical, really. It just lacks a pigment in the hymenium. It's just sort of brown. You add KOH, it stays brown. And another thing with this that has made it difficult is that you might have noticed back earlier in my slide, I have these 3 groups, these 2 that are closely related to each other and this other one. And now it might be nice if the purple one was this one off on the edge, but it's not. It's one of these 2 groups that are closely related to each other. So these other 2 species that so far I can't tell apart aren't even the closest related to each other. So it's not really truly a complex. It's just 2 species that look the same so far. And nevertheless, I'm able to assign at least one name to this group. So this will be hopefully in a publication soon. Saria coeloplata, based on a name from Norway, Biaterella coeloplata, that was published in the 1800s. So moving on from this relatively small group, we can go to the orange ones. So this group around Saria resini is going to be Zithia now, which is another really old name. So it's not a new name I've made up, it's from 1825. And if we look at Zithia in sort of an expanded tree with all these ITS sequences, especially the environmental sequences, really there's a lot going on here. So this breaks down into at least about 13 clades plus a bunch of singletons that don't really belong anywhere else. And probably these each merit species status. But again, the same problem comes up as we were having with the Saria deformis species. Oftentimes, it's really hard to tell, but you'll have one specimen, you think it's one species, but there are 2 of these growing at the same time. And I actually have proof of this in this case, because I've sequenced the same specimen from the Boston Harbor Islands twice and had it come out in completely different groups. So this is pretty frustrating. And if you look at them, you can see some differences. So I'll go through some of these excellent micrographs that Luis Quijada has taken. And one of the most unique ones in this group is this single specimen from really high up in the mountains of Arizona. The apothecia are really dark, almost red. And if you look at the assayi, in addition to the typical wall, they seem to have this outer additional wall layer. So really, this is probably a good character for this to say that it's a species. Unfortunately, this is only known from one collection so far, and it's not really in a location that, especially now, I'm in a position to return to and collect more of. But if we look at other ones, we can see other differences. So the cells in the outside might be really big in some species, they might be really narrow in others. Where color is located throughout the apothecia might be different. You might have a lot of orange coloration in the subhymenium, or you might not. The apothecia might have a little stalk, or they might not. In this case, this species seems to have a slight iodine reaction in the excipulum that doesn't tend to happen in others. And there are also species that, yeah, again, just some have a really long stalk, some don't. The asci for some seem to have this kind of structuring in the tip, but not in others. And really, otherwise, morphologically, the asci themselves, the paraphyses, a lot of the classic micromorphological characteristics are pretty similar. And with the fact that presumably some of these have got mixed up, it's really hard to tease them apart. So plans are in the future to really be a lot more careful about this. So if I'm looking at an apothecium, it's that apothecium that I will have either cultured from, or that apothecium that I will have sequenced maybe half of, which is kind of tricky because these are really small. At biggest, about 1.5 millimeters, 2 millimeters, but often more like 500 microns wide. So anyway, so this third group, this completely new genus, looks kind of similar to Saria deformis, actually, right? It's black apothecia. They have a stalk, which is a little unusual for Saria, the deformis group, the real Sarias. One of the big macromorphological differences is that you've got this layer of sort of crystalline matter, these pruina on the surface. Also, internally, the asci are much longer than either of the other two species. It has a similar iodine reaction in the asci. It also shares this characteristic that we have noticed seems to be a feature of this class, that the asci all have this not quite bitunicate dehiscence method. So the inner wall of the ascus will extend a little ways in the other species, and this species quite a long ways. And the spores will get ejected through a channel that opens in that. So this is kind of bitunicate, but maybe more, speaking technically, rostrate. It's a little hard to tell the differences between these sometimes. But looking at this, you might say, well, this just, I mean, really looks like the other ones. There are some differences, maybe. I mean, it looks different. But why maybe do these need to be 3 different genera? And one of the reasons why we've made this decision, I mean, there are molecular differences, and there are some morphological differences. But one of the big differences is that if you look at the anamorphs that are related to this species, what we have is a completely different type of anamorph. So before, we had these almost like little parathecia, but that produce conidia instead of asci. The closest related anamorph to this one doesn't do that at all. It's just naked asci, or naked hyphae, that end up fragmenting into these little cylindrical sections. So it's this arthric type of hyphomycete. And this, again, was placed in a different genus than either of the 2 other types of anamorphs. So this is part of what's informed this decision. Really, these anamorphs aren't alike at all. And really, this isn't even a type of anamorph that was ever previously known for this group. So its presence in this new genus makes me, or in this, or closely related to this new species, makes me kind of think that maybe these are better treated as genera, not so much as just different species or different clumps in a single genus. So another interesting thing that we've been able to do with this group, now that we've collected all this data from all around the world, is we've actually been able to do some dating analyses for these. And actually compare it to when we know that certain conifers arose. So one of the interesting things is that really this whole group, Sauriomycetes, we can put this as arising kind of at the same time as a lot of the initial diversification of Pinaceae and Cupressaceae, these two host families that it grows on, or also simultaneous with the diversification early on of the genus Pinus, which is one of the biggest hosts of this group of Sauriomycetes. And we can also see that a lot of these speciation events and a lot of these events of these different genera seeming to arise seems to happen at the same time that a lot of the other smaller conifer genera in these families that these grow on were diversifying. So this is a pretty nice picture, and this is, you know, a fairly old group of fungi that seems to have diversified basically as soon as this ecological niche existed. Which is a pretty nice picture, and it's something that hasn't been demonstrated before because there really just hasn't been enough data. So this is about it in terms of what you're really likely to find in the Northeast. But I also have some interesting things that maybe people could keep a lookout for, either because they've been recently described or because, well, they're all underreported. But there are just a bunch of interesting things from other parts of the world that could be here, or there's not really a reason why they shouldn't be here. So I'm going to start with, and I don't know how we are on time, but yeah. One of the more interesting entries has been, I think I mentioned, this group Orbiliomycetes, where there were about 7 Resinicolous species that were described just last month by Hans Otto Barral, a German mycologist who is basically the king of Orbilias. So he finally published his big monograph, hundreds of species, and these were among them. So one of these interesting ones that would be really interesting to find here is this beautiful genus Lilopila. So these are these beautiful violet apothecia that grow on resinous wounds. And so far they're only known from Europe, only known mostly in alpine areas. But they're really gorgeous. It would be really interesting to find these around here. Another group that was recently described is this genus Amphosoma, again in Orbiliomycetes, again described last month. And again, this is something that I think there's one report of one of the Amphosoma species from the Rocky Mountains. So they should be in North America. And for all we know, they could be around here and just haven't been picked up ever before. So this would be another thing to keep an eye out for, these little Orbiliomycetes. And there are a few other species. I'm not going to go over all of them. But yeah, if you have any questions about information about what things to look for, you can always contact me. I will be happy to talk about Resinicolous fungi. So another species that's around is Pseudocybe resini. So this is a relative of the other species of Pseudocybe that I mentioned first. This is originally described from Europe. There's also a fungus that looks exactly like it pretty much in the Pacific Northwest. Might be the same, might not. The conidia for these tend to be a little more rounded, not so oblong. but otherwise it looks pretty similar. So this is another of these hyphomycetes. There's a little fungus that a collaborator sent me from, two separate collaborators actually independently sent me from Europe, that seems to be something completely novel in Uroshiomycetes. So they look like these little green parathecia. And inside the asci are these big polyspored asci. And so far this is only known from Spain and Norway, but there's not really a reason why it shouldn't be here. So it would be another interesting thing to look out for. In addition to all of these fungi, there are a variety of hyphomycetes, which I really haven't dipped into very much in this talk. I've talked about the Cerasibes and some things like that, but there are really a ton of these. I would challenge you, if you have a microscope, just pick up a piece of resin and just start looking at random bits of it, and I can almost assure you that you will find some sort of fungus on it. But these really haven't been very well characterized. Some of them are known and described, some of them aren't. So there are a couple species of what were originally described as torulas that look pretty similar, that were known to grow on resin. There are a bunch of undescribed things that people have just looked at or shown pictures of. You can see these growing even over the sarias. I very frequently see black, sort of dark hyphae overgrowing them, and not necessarily parasitizing them, just growing around them. And these just need a lot more work. People need to start paying a lot more attention to these. There are also other apothecia. So here's one that's sort of a bluish green, and currently it's in the genus Clausinomyces. This species is Clausinomyces kirsteinianus. And it occurs on resin, little blue-green apothecia, quite pretty. Very few reports in Europe and no reports outside of Europe. Another one that I personally have seen, mostly in the Pacific Northwest, are these little slightly hairy apothecia growing on, I think, mostly spruce resin, that really seem like they belong in the genus Ciliolorina. It might be a described species, but it might not. It's just not really clear yet. And another group that are interesting and are really not understood at all is this family Bruceomycetaceae. So nobody knows really where these fungi go. There are only two species known that have kind of been put together based on morphological arguments. There's this Bruceomyces castoris, which is named after beavers because it was found on beaver scars in the Pacific Northwest. And its closest relative, supposedly, is this Resinogalia humboltensis, which is only known from New Caledonia. So these are, again, these sort of pin lichen type structures. So these are apothecia at the tip of a really long stalk. And people just don't really understand these very well. But they have pretty similar micromorphological characteristics. These almost like red blood cell shaped spores, which may be ornamented or not, depending on which of the two species you have. Not that you're really going to confuse them with each other, since one of them is from New Caledonia and one of them is from the Pacific Northwest. So there are more species, a few more species, definitely some really old ones that aren't known in the modern day very well. Some undescribed ones that people have seen. There's just really a lot going on here, and there's a lot of work still to be done here, even in the species that people think they know really well. So at this point, I want to thank various people. So for different points of this project, the National Park Service, the Friends of the Farlow, the New England Botanical Club, and Harvard's Department of Organismic and Evolutionary Biology have provided funds for this. I'm I'm also really grateful to all of my collaborators and also just the people in my lab. So I think several of whom are here: Don Feaster, Luis Quijada, Danny Hale Waters, Jason Kirky, and Jason's definitely here, so is Don. Kathy, Michaela, the various collectors and also collaborators that I've had in other countries. So Isaac Garrido Benevent in Spain, Peter Johnston in New Zealand, various other collectors, all the herbaria that have loaned me material to look at, and also all of these different bodies that have provided me permits to actually collect things and actually try to figure out these problems. And also my my partner for being there through, especially this past year, and helping keep me sane. I'm done.
Thank you. Thank you, James. Will we be having question and answer at this point?
I think so.
Yeah, my mouse just died for a second. I'm so sorry about that. That was wonderful.
Thanks,
you.
James.
Thank
That
you
was
so
great.
much. So the first question that we have is from Susan. Would you like to unmute yourself and ask your question?
Let me remind myself. I'm coming. I live in Idaho, and I'm about I don't know, 6 miles from conifer forest. Easy to get to any time of the year. Really, we have a ski resort about 15 miles north of Boise. So my question is, you referred to studies about what you're finding in the forests of the Northeast. Are there similar ones that will describe what we can find here in the Pacific Northwest?
So that's a great question. And actually there, with the exception of this one Italian guy in the 20s describing what he could find in his area, there aren't really a lot of comprehensive studies linking things together. In terms of what I know to be present, there are a bunch of different species in the Mycocaliciales that are present in the Pacific Northwest. So there are some keys to those groups. There's definitely like the different groups in Sauriomycetes are there. I've sequenced specimens of the orange ones from Idaho specifically, but also just from neighboring Washington. I have the new genus that I'm going to describe, and also some specimens that look like the deformis group, like the core Saurias. But really, ultimately, there aren't a lot of really specific local floras. Maybe the best treatment for the Mycocaliciales in your area would be, I think, in Bruce McCune's Microlichens of the Pacific Northwest. But there aren't, it's really smatterings of papers. It's not like single collected lists of what you're going to find. Yeah.
Well, I guess I'll ask my other question just quickly. Do you have to climb trees to get at the resin that will show you the fungi?
So for big trees, it's really hard to find these sort of pockets of resin on the outside, but on dead trees and on younger trees, not really so much. I have never really climbed a tree. I think the most I've had is to get something higher up in a tree. I think our postdoc Luis got on my shoulders once to try to get something. But I've done most of my collecting not at above head level. So
this is a good argument for having the local club foray, plan ahead, take a ladder.
Yeah.
And it's not like we're gonna find drops of resin on the ground or something and pick it up.
Yeah, probably not. I have seen one specimen with resin on a fallen pine cone that had some fungi growing on it.
Wow.
Yeah, but mostly what I've seen is still on the tree, so not really like fallen on the ground, growing there type stuff.
Thank you.
Sure.
Great, and the next question we have is from Maricel. Maricel, would you like to ask your question?
Can you find these fungi at any time of the year?
Yeah, as far as I can tell, it really seems like they're not too picky. So Sarias, or that group, the Sariomycetes for sure, you can find them any time of year. I've found them in spring, I've found them in autumn, I've found them in winter, I've found them in summer. There other ones like the Eustilbum definitely are present, at least in summer, and I think also in winter. A lot of these are so scant that it's kind of hard to really say if there's a seasonality to them, but they seem to be on this pretty dry substrate, so they seem to be more the type of things that stick around and then rehydrate rather than growing for a very, very brief period of time and then decaying.
Okay.
So I think you're probably good to find these almost any time of year. Yeah.
Okay, I'm going to the woods tomorrow.
Good luck.
To the pine barrens. I get two more questions. Should I wait or should I ask now?
Go for it, Maricel.
Okay, so I have another question is, are the Sarea species fruiting bodies connected? Because,
So,
yeah, go ahead.
so do you mean like, are they connected by a mycelium inside the resin?
Yeah, some kind of running like,
Yeah, so there, you can actually, if you take a pretty good section of these, you can see a mycelium inside the resin spreading around. So they do seem to be connected. And I think there have been studies that have estimated that the mycelium for a single species can be, I think, like a meter long in a tree. But again, a lot of the old work on these have been done assuming it's all the same thing, so it's maybe not so clear in light of molecular results how big these individuals really are, but they do seem to have a mycelium inside that you can see in the resin. Yeah.
Sorry, I muted you, Maricel. There was just some weird sound, but go ahead and ask your last question.
okay. My last question, I am probably wrong or misunderstood what you were saying, but few times I thought you said hypomyces or
Oh.
hypomycetes. Why do you say that? I thought that I have hypomyces. Is this, you know what I'm trying to say?
Yeah.
Okay,
So what I was saying was hyphomycetes. So I'll just put it, I'll type it out in the chat.
I saw it. It has an H and mycetes.
Yeah.
Yeah.
So hyphomycetes are, it's just a name for a group of anamorphic fungi that don't really fruit inside a structure. They just make their conidia either at the end of a stalk or just on the surface or in little cushions. So it's just a name for a group of anamorphic fungi. They're not monophyletic, so it's not really a class. It's kind of a holdover classification. But yeah, hyphomycetes. So they produce synemata or just naked conidia or sporodokia.
So
Yeah.
the hypomyces, the hypomyces that we usually find, the ones that are bigger because are growing on bigger hosts, like the white cottony stuff.
Oh.
Not the same. That's what I'm confused.
No, so this is, yeah, I see why it's confusing, but they're not actually related at all.
Okay.
Okay, great. And then the
Yeah.
next question we have is from Jason.
Me?
Yep.
Oh, okay. I think I had asked about cultures. Do these grow in culture?
Yeah, so the various Saria or Sariomycete species do seem to grow in culture. People have even gotten the anamorph in culture. I've not succeeded in doing that. But those can grow in culture. The Eustilbum I've been able to culture. It grows really, really slow, which I'm sure you're familiar with, Jason. In terms of other things, the Clausinomyces olivaceus, that'll grow in culture. So there is sort of something interesting there where these things seem to be happy growing even when they don't have the resin, but for some reason in the wild, you only ever find them on the resin. So there's definitely something interesting there. They're capable of metabolizing whatever's in PDA or CMA, but they only really grow on this really inhospitable environment. Yeah, it's a great question.
Yeah, I have another quick question.
Sure.
So are all of these, the Saria and their allied taxa, are they all polysporous? And do you have any literature on the kind of biology of polyspory?
So they do seem to be. Everything in Sariomycetes that we have a known anamorph, or sorry, a known teleomorph for is polysporous. And it seems like truly polysporous. You can also see, or in some of the other photos, that some of these other things are polysporous. The Clausinomyces olivaceus is not truly polysporous, but essentially shoots out bundles of tiny spores. This unknown Uroshiomycete is polysporous. So in terms of why that is, there's a Martha Sherwood paper that just sort of notes that this seems to have occurred a lot on these in these groups of fungi that grow on these really xeric environments. And I think there's also a paper that was published in 2004 specifically that looked at polysporous lichens that tried to see like how this characteristic had arisen, this polysporous characteristic, because you find it a lot in different types of lichens that aren't necessarily related to each other. So there definitely seems to be something to it, but why doesn't necessarily, because I believe in Martha's paper, she also says that some of them go completely the opposite direction and put all their effort into single huge spores too. So yeah, why this is a good strategy, why these extremes are good strategies for these substrates is, I think, an open question.
Thank you.
Yeah.
And Ethan,
Great talk. Thank you, James. My question was about what component of the resin these fungi are eating. I mean, I guess we can assume they are living on the resin and they're eating the resin. And also whether they've evolved enzymes that are significantly different from the wood decayers.
Yeah, so these are great questions too. So to my knowledge, it's not really known. Some of them seem to be a little specific in terms of what they'll grow on or what they'll like. So one study I'm aware of was done in Japan back in, I think, the 80s, where they tried to grow their Saria resini on the resin from the tree that it was found on and also from other species, and found that it seemed to grow best on the resin of the tree that it was found on, in that case, a Hinoki cypress. But as to why that is, it's not really known. In terms of them producing enzymes that are distinct from wood decayers, this is presumably the case, but to my knowledge, nothing's been published conclusively that is looking at that. There might be some people working on getting genome data on that could say more conclusively, but that's also something that hasn't been, well, if it's been answered, it hasn't been published yet. Yeah, and the question of what they're eating is also just not really known. Again, they can frequently grow in culture, so whatever they're eating doesn't seem to be necessary for their survival, but yeah.
Great, thank you.
And the next question we have is from Max.
Hey, thank you for your talk.
Yeah, sure.
So early on in the lecture, you mentioned that these fungi are primarily higher ascomycetes. But what else does that yield? Sorry, I'm holding a pierogi. What else does that yield? Like, are there lower ascomycetes or basidiomycetes that also inhabit this environment, or is it just anyway?
Yeah, so there are things like yeasts that grow. I think there's at least one species of yeast, I don't know the name off the top of my head, that does seem to grow in resin. There's also a species, Alicidium resini, that where it belongs is kind of not known. It's an anamorphic fungus, but at last placement, it was considered an anamorphic basidiomycete. It doesn't seem to be specific to resin, though. It'll grow on it and off it. So frequently, I mean, when it was originally described, it was on a dark resinous wound, but a lot of the subsequent reports of it are not. They're just on rotting wood. So what that means is not very clear. So there are some, but really the bulk of the ones that are really well characterized or are recognized as primarily growing on resin are these higher ascomycetes. Yeah.
Okay, cool. Thank you.
Sure.
And we have a question from Ari, but I wonder if it was already answered. Ari?
Wow.
He wrote, how are these microfungi obtaining nutrients from the lipid soluble terpenoids?
Yeah, again, just not known. They do genuinely seem to be, as I said before, they have they have these hyphal networks running through the resin. You can even see in some of them, I know it's been reported for these Mycochlyciales, you can see where the hyphae have actually kind of hollowed out passages in the resin where they've apparently eaten away at the stuff around them. But how they're doing it, just not known yet. Yeah. What they're eating, why they're eating it, just, yeah, open question. Yeah, so yeah, even really basic questions like this for this group of fungi just don't have answers yet. So that's part of why I think it's just this really interesting group.
Great. I'm just going to open up the floor and ask if anyone else has any questions, to just go ahead and unmute yourself. Well, thank you very much, James. That was awesome.
And NYMS members, at some point when the pandemic is over and when we're able to do things in person again, we're going to have James down for a weekend to do a microscopy workshop with us, and we'll go on a walk or two to places in New York City where we have pine, and we'll really scour the area and see what we can find. So that's something to look forward to.
Yeah.
Excellent.
And yeah, meanwhile, if you go out there and start looking at your pine trees, you will find these things. Since I was introduced to them by James last year, we found Saria resinae, I think, in all 5 boroughs. and we found Lachnellula resinaria. So these things are here in the city. So I urge everyone to go out and start looking for them. They're very exciting to find, I might add.
I have a quick question. So it sounds like it would be useful to carry around like a loop or something. Can you hear me?
Yeah.
Yeah, so I mean, most of these things are pretty tiny, right? So
Yeah, they're pretty small. Nothing's huge here. Yeah.
so like I imagine you're probably carrying a loop around or magnifying glass or something.
Yeah. So I generally take a loop. Another thing I would recommend is not only a loop but a light. So like a headlamp or either that or like a lit loop, because it's really it doesn't really help when something's stuck to the tree. You have the shade from the tree. You have the shade from your head trying to look at it.
Right.
So yeah. I would recommend a loop and a light. Yep.
Huh.
And
Sorry, but James, when we were out with you, you had a spritz bottle and
Oh yeah.
and paper towels, I think, to keep the thing moist.
So yeah, I had a spritz bottle. Yeah, if you want to look at things alive, most of these, even when they're dried out, they're not really dead because again, they seem to be these fungi that prefer these xeric habitats do this. They can dry out without dying because they don't normally have a lot of water. So if you want to look at them alive, you can spritz them with water, keep them in a moist chamber, and you can get them to shoot spores and you can culture them. On the other hand, since they're used to dry environments, if you wanted, you could probably just collect them dry and rehydrate them when you got home too. But yeah, for other things, more sensitive things, a spritzer bottle and plastic bags are helpful to keep things alive, like delicate things like orbilias that would maybe die if you dry them. It's handy to keep
Also,
them.
I think you mentioned, I think you told us that it was easier to find them when you
Oh
get home.
yeah, that too. Things just get bigger when they're things just get bigger when they're full of water, so it's just easier to see them too. Yeah.
I actually have one more question.
Mhm.
To me, I mean, it was very interesting to listen to your talk. So thanks so much. And I guess I know a little bit about lichen. You know, they grow also in some pretty odd places, right? Like on the surfaces of rocks and like even in like really inhospitable environments. So
Yeah.
do you think, is there some sort of evolutionary relationship between the fungi that occur in lichens and these resinicolous fungi?
So in terms of a really close evolutionary history, not really. But that being said, the fungi that occur in lichens and themselves don't all have really close relationships to each other.
Okay.
So some of them are really like Arthoniomycetes. That group is really closely related to the Bitunicate ascomycetes. You have things like Lecanoromycetes that are off with their big group that's closest related to Eurotiomycetes. And then I think also Coniosabomycetes, some of these pin lichens are one of these kind of basal groups that aren't really related closely to anything else either. So there's not a really close evolutionary history with any one of these groups, but it also seems clear that the lichen habit evolved multiple times independently. So in the same group that this is in, just the general big group of higher ascomycetes minus orbiliomycetes and pizizomycetes, but yeah. So not really closer related than that, but
okay.
Yeah.
Thanks.
So it's kind of hard, yeah.
Thank you.
James, I have a quick question if that is all right with you.
Sure.
Thank you so
Yeah.
much. Fantastic presentation. Following Misha's first question, I was wondering what microscope or what materials does one need to obtain detailed views of Conidia and asci?
Yeah, so you're going to need a compound microscope for especially like small spored things. You probably need a 100x objective.
Okay.
Yeah.
And would you need that that sort of microscope oil?
Yeah.
Okay.
Yeah. I mean, you could probably see the asci without it, but it helps to see fine features. Yeah.
Okay. And does one need a slide for that then? And
Yeah.
a cover
Yeah.
slip?
You'd need glass slides and cover slips. Yeah.
Okay.
And in terms of stains or mounting media, water is like just tap water is good. KOH is important. I think Luis, Congo Red is also helpful, which is not maybe that easy to get. And for staining an asci, the Meltzer's or Lugol's iodine, yeah, also useful.
Great, thank you so much.
Sure. So I think I see two other questions in the chat. Someone asked, or someone said that it looks like Saryomycetes evolved at the same time as Cupressaceae and Pinaceae. Do I have any thoughts on how that came about? So at the moment, the primary thought I have is that that should be about the time when conifers evolved this ability to produce large amounts of resin. Because these are really the groups that do that, minus Oricariaceae. So it just seems like this is probably about the time that that niche would have appeared, the niche of just having big blobs of resin from conifers. And it seems like maybe, I don't really have concrete answers to this, but maybe a group of fungi that wasn't doing so well on something else was able to live on this and just kind of stuck with it. I had a guess. So somebody else asked if the resin itself has any inherent antifungal purpose or property that these fungi have overcome. So this is a thorny questioning to answer. So there's been a lot of research on this that's mostly done in laboratory conditions, and there's a lot of debate about whether there's actually these laboratory conditions really translate to in-person, like in the field conditions. So there have definitely been studies that show that if you grow fungi in air saturated with a lot of these single compounds that are found in conifer resins, it does inhibit growth. On the other hand, people have said at the surface of resin, most of those go away because they just evaporate off. So maybe it doesn't matter so much. So, and the other compounds, the more solid compounds, since they're not really volatile, maybe they don't do so much. Certainly mechanically, resin forms to catch things in it. I mean, it certainly does that to insects, just kills them by drowning them, basically, covering them in a sticky coating that they can't escape. It also just closes up the wound, and presumably, I mean, this is where the amber specimens of these Resinicolous fungi are from. These fungi seem to get caught and die in washes of resin. So there are mechanical, and it seems like there are chemical features that would make this a substance you really wouldn't want to grow on. But again, they seem to do fine. But as a caveat to that, some of these studies that have looked at single compounds have also found that fungi that really don't have a reason to be resistant to them seem to be fine with certain compounds that inhibit other fungi. So it's a big question. People have tested it with individual compounds, but it's hard to test in a sort of everything at once way. Yeah. Hemotoxin. I'm not sure I know what that is. So if hemotoxin. So what is hemotoxin?
Maybe you can ask.
Oh yeah, Ari, feel free to unmute yourself.
Oh.
Maybe his mic doesn't work or her mic.
Oh.
Oh well.
Yeah.
Well,
Anyway, in terms of actual toxins, as far as I know, none of these are poisonous. They're really small. They also, the ones that I was discussing here, don't really seem to be agents of disease in plants either. They seem to be mostly incidental, though there's kind of a question there too for some of these, like whether it's actually an agent of disease or whether this is just growing there on the resin. There have been various studies of pathogenicity with these Sareomycete species that have shown no, it's not actually causing disease. And then there are other studies that have suggested, well, maybe it's causing disease. So it's, in terms of actually affecting the plant negatively, my personal thought is that probably not, but it's a little bit unclear.
okay, great. Well, thanks again, James.
Great talk.
Yes.
Yeah, thank you,
Thank you.
James.
Thanks. It was a pleasure to talk to you all.
And I really look forward to seeing you in person.
Yeah, I look forward to seeing you in
in coming
person,
months.
just seeing people in person in general.
Not too picky. Anybody?
Well, thanks again, James. That was really that was great.